So the lecture number 3 on our crystallography course is going to be given by Meindert Lamers, and it's going to about some dark art, the expression, purification, characterization, and crystallization. Let's leave it.
So today I'll be trying to tell you a little bit about the things that I try to do to actually get a crystal that not only looks pretty, but actually will give you the diffraction data. But I was asked by Andrew Leslie to briefly show this. This is for the practicals. I haven't read it. Is there anything I need to add?
And the repeated symmetry lecture for those who couldn't make it on Friday.
And so the symmetry lecture will be today.
2 o'clock.
Tomorrow, 2 o'clock. All right. So the goal of today is you've got a bit of protein, reasonably pure. Then you do something magical, and then somehow you get yourself a crystal. This is, by the way, real. It's not protein crystal, though. And then you shoot some X-rays at it, and you get a beautiful diffraction. And that's the goal of what we're trying to do today. Well, not just in one day. But. So this is what I took from Jan Leuven's talk from last week. These are the different steps that you'll have to go through and that will be discussed throughout this course. And one thing I really want to emphasize is that most of it, or your work, will be, where's the pointer? Will be actually this. All right, so you come to the project, they tell you go work on that protein, and you think, fine. You have no clue that 2 other people already spent 2 years doing that and didn't work, so you have full optimism. And then they go and let you fill in the lab for a long time. Then you finally get those crystals. You go to a synchrotron, you have no clue what you're doing. And so there's a postdoc who'll probably collect the data for you, and if you're lucky, you may collect the data set as well. And then you need to phase it, and again, the postdoc is going to help you. And if he can't or she can't do it, then you probably just call Randy Reed or Phil Evans and say, I got a really difficult problem, can you help me? Again, then the model building and refine model, that's mostly the postdoc, and if it doesn't work, you talk to Garrett. And then finally you come to the end, and because you don't have all the knowledge, it's going to be the boss that's probably going to interpret the model. So this is the part that you're going to make the biggest difference in the project. And actually, I find it the most interesting and most exciting part of the whole project, because this is where you can really tinker with the protein, find out how it works, and find maybe that essential clue that will give you the crystal. The rest, not to trivialize it, but it's kind of straightforward. All the methods are in place. If you got your crystals, this can happen in a matter of days. But that can take you several years. Now, I'm going to talk about stuff that I know a little bit about, and there's many, many more things that you can do. So this is just my limited view of how I do crystallography. That by no means I'm going to tell you this is the only way, but I might be able to tell you a few things that make you think, oh, I could try that as well. So let's start from the very beginning. Somebody tells you go and crystallize, say, a ribosome. Where do you start? Well, you need to choose what organism will I start from. In some cases, that is incredibly diverse. You can take any ribosome from any organism. It might be a good target. However, if you want to be solving a particular protein that's related to cancer, you're very much limited to mammalian proteins. So that affects how you work. But what really matters is the diversity. This is a sequence alignment of about 50 different protein sequences, and all these different colors indicate different residues. But the point here is evolution has made an enormous variation of the proteins that are available. So you should make use of this. These will all look very much the same structurally, but they have little changes that can make a very important difference between having no crystals or having crystals, or having protein or no protein. So by saying I'm going to work on E. coli, or I'm going to work in Thermus aquaticus, because many other people have done it, you're limiting yourself extremely. You need to try as many different species as you can get your hands on, with a little bit of limitation. But there might be other reasons why you want to work in E. coli. Maybe there's an enormous wealth of biochemical data available that you want to make use of. So in that case, it makes sense to work on E. coli. But in many other cases, if it doesn't matter, if you just want to get a ribosome, you make use of many different organisms. So doing an alignment is very useful. and there's another reason why it's useful, is that it also gives you some information of domain boundaries. So you can see there's all these gaps in between the different sequences, which means that evolution has put some extra residues in here, or maybe removed a few, and that may give you some indication of that may be the end of a domain. So possibly if the conservation is a bit poorer here, maybe we're going to truncate it here and only express this bit, or the other way around. You can do secondary structure predictions, which are extremely powerful, not only at this point, but also during the building of your structure. If you have a total novel structure, having these secondary structure predictions can actually be very helpful to you when you're trying to build a piece of helix. And maybe the helix, especially at a lower resolution, say around 3 or 2.8 angstroms, it may not have all the features that you can really say that's an arginine, a lysine, or whatever residue. But if you use the information of a secondary structure prediction program, you can use that to say, well, I know that the prediction is from residue 12 to 22, so that's what I'm going to build. And I myself have been amazed about how accurate these predictions can be, and maybe off by 1 or 2 residues. So there's many different websites you can do that. I personally like the Max Planck Institute toolkit, which has all kinds of nice features. These alignments I typically do in Clustal, and there's many other programs you can use. And then I used GelView to actually visualize it and then manually edit a few sequences if you want to move residues over or not. Again, that's what works for me. May be something else will work better for you. But certainly staring at sequences like this can be very informative. Don't spend too much time in it though. Another useful thing to do, and this is something that we recently did with my student in the lab, Ulla, is that if you have a homologous structure, and for some reason you want to solve another structure that is related, you can use threading algorithms to mold your protein sequence into an existing structure. And that can be for a domain, that can be for an entire protein. And suddenly, this linear sequence alignment gives you structure information about a particular loop that in one protein is small and another one is much larger. Possibly it's very flexible, and that's not going to be very good for crystallization. So you might want to take that out. On the other hand, if this loop appears to be close to the active site, it may have a particular function, so you don't want to take it out. And that you wouldn't be able to really get out of a simple sequence alignment. So in this case, we used a program called Modeler, which was developed by André Sali at UCSF. Again, there are many other ways you can do it. But if you can, if there are homologs, even if it's just for a domain, try and thread it into in that structure, and you might learn a lot of things that you didn't know before. Now, you can either make use of nature and try all kinds of different organisms, but you can also look at your structure and make an educated guess on where there's a little bit of variation. And if I go back here, so you see that there's a bit of variation here, and if you're particularly interested in your protein, you might say, well, maybe I can get rid of that loop, or this one has 2 charged residues. And typically charged residues do not like to be in contact with another molecule in a crystal lattice, so they like to be sulfated by the water, and therefore they may not participate in the crystal contact. So by changing, say, an area which is exposed and maybe very variable in your sequence alignment to something that's more hydrophobic, you might create a new crystal contact, and that therefore gives you new crystals that you haven't had before, or different crystals if the ones that you are working with are not good enough. I didn't have enough time to find a program, but there's actually somebody who spent many years and has now a tool. Anna, is Anna here? No, okay. She would be able to tell you. So then you can, if you don't have any homologous models to do this, you can base simply on the sequence, predict which area might be suitable to change. And that, again, is varying the properties of the protein, not that you destroy the structure, but make it more likely to crystallize. On the other hand, if you know that you've got a catalytic enzyme that likes to, in this case, eat up DNA, you want to have a co-crystal structure, obviously you want to do something to prevent the DNA from being eaten up. So in this case, this is an exonuclease. They mutated this glutamate into an alanine, which allowed them to co-crystallize the DNA. Now, there's a very important thing is that kind of what I often say, you need to make your protein happy. So if you want to crystallize it and you've purified it to complete homogeneity, but it tends to be binding to partners or it has a special affinity for ligands, but you remove them all, then maybe you have a protein that is very unhappy and therefore will never crystallize. So knowing a lot about your protein is extremely important because it will give you the crucial ingredient to make it crystallize. So if you know about partner proteins, try and co-crystallize them. If there's a particular peptide you can find out about so that you don't have to do a whole partner protein. Obviously DNA or RNA if you have a nucleic acid binding protein, different ligands, or even an inhibitor. I know of a case where a group was working on CAMP kinase 2, which is involved in memory, a very large dodecameric protein with lots of floppy domains. And I think it took them 4 or 5 years of trying until they got crystals, and this was over a period of 3 people. And the crucial bit was that at some point, some other group had found an inhibitor of the CAMP kinase. And with that single inhibitor, they finally could get crystals of the protein. Now, this is an example of something that we did a long time ago. This is a DNA repair protein called Mutagenesis. And what we always did is that we, after the purification of the protein, we always standard did an ATPase assay to make sure that what we purified was the same as before. And we've got crystals of the native protein. We've tried for many years to solve the structure by different methods, and we completely failed at it. And at some point, we went to express selenomethionine protein. And suddenly we found that this protein was more active in the ATPase assay. Didn't really pay attention to it. But then when we grew the crystals of the selenomethionine protein, they turned out to be twinned, which you may learn later in the course is something that you really don't want to have. And it basically made it impossible to solve the structure. And so we were scratching our heads and wondering what to do about it. And at a group meeting when I presented it, one of the postdocs said, but hey, didn't you have high activity? Well, why don't you add some ADP to the crystallization conditions? And adding a single ADP resolved the twinning and allowed us to solve the structure. And after 3 years of struggling in the lab, we put the crystal on the beam, we went for dinner, came back and the structure was solved. And it was actually a little bit disappointing that after so much work, it was so easy to do it. But the point is, if you try to understand your protein, you will find the secret ingredient that will get you the crystals that you need. But sometimes you really don't have a clue. So at some point, I was working on a DNA polymerase, and I still am, and I wanted to co-crystallize with the DNA substrate. And I look back in the literature about how people have gone about and get co-crystal structures of polymerases with the DNA substrate. And this is just a list of a few, there's many more. And what I've highlighted is in black is the piece that was actually seen in the crystal. And in red was the sequence that was present in the crystal, was present in original substrate, but within the crystal you could not see it. And then in addition, many of the conditions added extra nucleotides, dideoxynucleotides, that would be incorporated into the crystal or before crystallization. So there's no way you can sit down and think about it. I'm going to use 24 nucleotides and a 3 residue overhang, and that will give me crystals. Here, in this case, this is pure luck, especially if you think about this one where you have half of your DNA substrate not even visible. Logically thinking, you would never have done it. In some cases, you just don't know what to do. Well, there's one thing, and I hope I'm not going to offend anybody, but what I don't like is doing limited proteolysis. There may be a few anecdotal stories that it may have worked, but just think about it. You use a protease that's going to cut a particular size and rip your protein apart, and what you're left with is a band that may be stable, but you don't know what the ends are. And likely it have cut maybe smack in the middle of a domain, so now you've destroyed something, or it may be in a long flexible loop and it's cut in the middle and you're still left with a 20 residue flexible thing. So it doesn't really help you. And maybe, yes, there are a few nice cases that it worked in, but I wouldn't spend much time on it. Plus, if you want to do this and purify it, you put the protease on a column and really make your colleagues upset. Then the other thing is, and I find this really amazing, is a lot of time is spent by biochemists chopping up the protein, attributing all kinds of activities to domains that are completely messed up, and they publish a structure, and this is how it works. And then if you solve the structure, you find that the actual structure has nothing to do with whatever they predicted. So if you go and base your work on somebody else's work and say, well, it must be true because it's published, you're going to make your life really, really difficult. You can use it as a suggestion. Okay, so there's an active site which is actually completely somewhere else, But maybe it's there. And maybe there is some evidence that there's interaction with a protein called beta somewhere. So you can take that, but go and find out for yourself if it's true. Don't believe what other people have printed. I'm absolutely useless at cloning. All my people in the lab are super at cloning. So for that reason, I really like this method. And there's many other methods, but what I don't like is restriction enzymes. I once had I struggled a long time with a particular cloning that was in my PhD. And you know, the typical way, restriction enzyme, you cut your ligate and the whole lot. And after 2 weeks of trial, I was having a group meeting where I presented this. And then somebody said, well, which tube did you use? Well, the one there in the freezer. Oh, no, that one. You should use the other one in the other freezer. So the fact that you have all these restriction enzymes, and when you look at them, the expiry date is 2004. It just doesn't work. So we've been using a method that relies on a single restriction enzyme and a single enzyme called T4 DNA polymerase. And maybe many of you are familiar with it. I had a cartoon, but I lost it. But it's very simple. We have a set of vectors which have the same insertion sequence. And the trick is that there's a long stretch of DNA, say 10 to 15 amino acids, nucleotides. And there's only 3 out of the 4 nucleotides. And when you treat this with T4 DNA polymerase, which has an exonuclease domain and a polymerase domain, the exonuclease will remove a stretch of DNA until it comes to the place where the 4th nucleotide needs to be incorporated. And if you do this in the presence of that 4th nucleotide, the polymerase will put it in there. So basically, the exonuclease will remove a piece of DNA of 10 to 15 nucleotides, and then the polymerase will make sure it doesn't go any further. You do exactly the same with your PCR fragment, and that means that now you're ligating long pieces of DNA that will anneal very, very efficiently. You don't have to use a ligase to anneal it, but close to Nick, and you can just transform it. And the other beauty of it is that you do a single PCR reaction and you can stick it into 15 different vectors with different antibiotic resistance tags or whatever you like, even for different expression in different systems. So that's what I like to do. There may be other ways of doing things, but I like to keep it simple. Okay, that wasn't supposed to be there either. Anyway, the point is we all kind of rely on methods that have been developed a long time ago. So when we typically do protein expression, you grow your bacteria for a bit, and then somewhere around an OD of 1, they leave the log phase and go into stationary phase. Now, there's actually something really cool going on. It has to do with invasion of a host, protecting itself, and there's biological warfare going on, but that's not really important now. What is important is that they stop growing and they completely change their metabolism. So what is important to know is, well, what's causing this? Why do they stop growing in the log phase, and why do they reach a plateau of, say, an OD of 1, maybe 2 if you're lucky? Well, it's actually really simple. You can get to much higher OD levels, and I've been getting 20 or so, just in a simple culture flask. And that was because Bill Studier from Cold Spring Harbor, who developed originally the T7 expression system that we're using now, in IPTG induction, all the PET vectors, was developed by him. I guess somewhere in his pension, he had a lot of time, and he literally went to every single ingredient that we use in a medium and find out what is really crucial. So typically what you do, you go to the kitchen, you get a bottle, and you start your growth, but you have no clue what's in there. You can even make your own medium, but you still don't know, because what you use is just a yeast extract or a peptone that you buy in a bucket. You don't really know what's in there. So this is a very, very long paper. It's worth a read, especially if you're tired and you want to go to sleep. But the main point, I loved it. He went through every single point, every amino acid, every type of buffer, every pH, what you could think of. But in the end, he said, well, what if you just take a rich medium, so 2 times TY, which is what we have, or terrific broth, which is not commonly made in the kitchen, and you add a little bit of magnesium sulfate, you can get to ODs of 18. And that's in a normal shaker flask. So that's what we do as well. We take 2 times TY, add a bit of magnesium sulfate. But importantly, we also use these culture flasks that have the baffle, so these indentions at the bottom, which give a much better aeration. They're also slightly shaped different. And then the glass flask, which you give it much better aeration. So recently, the autoclaves in the kitchen weren't working, so we ran out of these, and we had to use the glass flask without the baffles. And we couldn't get to the same densities that we typically get, so we got about 4-fold reduction in the amount of cells that we get. So the simple trick is you need to give them a lot of food. So LB is not good, it will run out of food. Magnesium is necessary because it's one of the most used metals in all kinds of enzymes. You need a lot of oxygen, which is why fermenters work really good, but the fermenters are, of course, very difficult to work with. It takes you 2 or 3 days to get your cells, etc. These are dead easy to work with. Then if you have a protein that may be a little bit toxic to your cells, you want to prevent any leaky expression, and that you can do by adding another plasmid called P-lyse, which prevents leaky expression, or simply by adding a little bit of, that should be glucose, sorry, not glycerol, which also prevents leaky expression. And then one other thing is that I found out for some of the proteins is that if it's toxic, the cell will try anything to get rid of it. So often it will remove the plasmid from your culture. So what I really do not recommend is to start in a small culture. So you transform your cells, you pick up a single colony, you do an overnight starter culture, and then the next day you grow up your cells, and after a while when they reach an OD, you start inducing. Well, at that time, your cells have gone through many, many, many duplications, giving the cell the chance to evolve and either mutate the plasmid or get rid of it altogether. So by the time you're inducing your cells, most of them have lost it, so you get much less expression. So typically what we do, that's another point, but what we do is I transform enough cells so I can plate it up on 6 plates. That's overnight, and it's a very stringent selection on an antibiotic medium, and so the cells are not likely to get rid of the plasmid. And then we scrape all the cells from all the 6 plates and use them to inoculate 6 times half a liter. You grow it for 2 or 3 hours to an OD of 3 or 4 or 5, and then you start inducing, and you only induce for 2 hours or so. And that means from the moment that you transform to the moment that you harvest your cells, it's actually a very small amount of time, so they've only been through a few doubling rounds, and therefore there's much less chance that you get loads of protein. Another thing is that it may be easy to grow up your cells during the day, and then in the afternoon put them at 16 degrees, put them in the shaker overnight, and then the next day you come in and you got lots of cells. But that's absolutely not necessary, because this is again from this paper from Bill's studio. This is using auto-induction, but when you use IPTG to induce, you get similar results. So from the moment that you see induction starting, within 2 hours, you reach maximum protein per cell. And if you continue, that's not changing. So what you're doing after 2 hours is you're not getting more protein per cell. All you're doing is getting more cells. So why not just change it around? First get a lot of cells and do a very short protein expression. Because otherwise your protein is sitting in the cell for 16 hours, well not all of them, but many of them, which especially if your protein is prone to degradation, it will be chewed up to pieces in the cell before you even get to lyse it. So then any protease inhibitor that you're going to add to your lysis buffer is not going to make any change because your protein has already been degraded. So this is crucial for us in at least 2 proteins that we work with, that we do a very short expression, even 1 hour, so we don't even reach full activity. But that way we can prevent any proteolysis of the protein. So what we then typically get out of 6 liters of medium, we get up to 100 milligrams of protein, and that's enough to set up the 20 plates that we have available. We could go to even more, but currently we're just flooding our columns with protein anyway, so we end up throwing away protein as it is. So I have no clue about protein expression in yeast or baculovirus or any other systems. But kind of what I've been trying to say in the previous few slides is that you should never think or even say, but this is how everybody does it. It really gets me worked up if somebody says it when I ask them, why do you do it this way? Well, that's how we used to do it in our own lab. That's not a good excuse. And these are just a few examples of how people have improved existing methods to do something completely novel. So Andrew Carter has been growing yeast to ODs of 80 and getting lots and lots of protein. Imre Berger has been using the baculovirus system to get, up to 10. How many proteins can they stick into one cell? Many. That should have been Wayne Hendrickson. He came up with the idea of how to introduce selenomethionine into protein expression that can be used for phasing of crystals. Surely at the time he began, people said, well, you can't do that. but he went ahead and did it anyway, and now it's the most commonly used method. And then I mentioned already Bill Studier, who actually, probably he was a microbiologist studying phages and realized, well, actually we could make use of this. So a phage that tends to invade an E. coli and then possibly lyse it, if you mutate it a little bit, you can make use of the system and then get protein expression like the way we do it now. Okay, protein purification. I really like this bit. I think this is the coolest bit because you can do so many different tricks to get to your protein. There's no standard way of getting to your protein. Many of you will just get a his-tag protein and bang it on the nickel column and come off, and that's it, done. But in some cases, that's actually not a good idea. Maybe it doesn't work, or maybe you want to get rid of the his-tag, or maybe the metals that also come off the his-tag, all the nickels, they come off and actually affect your protein. Might not be good. So there's lots of other ways that are, some of them are ancient, some of them are more recent, but I'm not going to go through all of them. And there's really good books on the GE website that take you through all of them. So it's worth a read because they explain a lot about the principles and the practical approaches to do this. But I do want to say a few words about tags, and we just use them because everybody tells you to use them. But there are some restrictions to it. One of them is that, for instance, a GST tag is great because it gives you extremely high purity, but it's also a dimer. So when you then want to characterize your protein and you say, oh look, it's a dimer, well, not so good. In some cases, it can be good. This is dynein from Andrew Carter, and dynein is normally a dimer, but in this case they removed the bit that dimerizes it and replaced it with GST, and so they can actually have a functional protein by using GST. This is an example I heard about at a talk last week. The structure is not published yet, so I can't get the real structure, but the point is, in the middle of the protein is the active site, and in the center of the active site is the N-terminus, and it's part of the active site activity. So if you stick a fat his-tag in the middle of it, you completely mess up the protein. Plus, they weren't able to cleave it off because the protease site was buried in it, so they had to denature it under mild conditions to actually get the his-tag off. So you have to be very careful in these things. Obviously, if it works, go for it, absolutely. But if it doesn't work, think about it. Maybe you shouldn't be using a tag, or place it at the other end, or maybe completely remove it. It's not so difficult to purify your protein without a tag. It can be done, especially if you play a little bit with the conditions. And then not to forget that, as I said before, if you use a metal column, there will always be metals that come off and will sit in your protein. And if you have a protein that doesn't like nickel, because it's a zinc metalloenzyme or whatever reason, you change its behavior. And if you then want to characterize it, you also get all kinds of different results. So again, a good reason to think about what you're actually doing. Okay, I skipped a few, that's right. And size exclusion, it's kind of, the end of my purification. I put it on the gel filtration column. Look, I get a single peak, I'm happy. But then it's very diluted and you need to concentrate it before you can freeze it because it won't crystallize at 1 mg per mil. You need to go to 10, maybe 20 mg per mil. You stick it in one of these horrible devices that will completely mess up your protein. And the reason is that you got 15 mils of protein and all the concentration takes place here at the bottom. It doesn't really show you very well, but you know them all. These are these conical tubes that have a V shape. And so here at the bottom, your protein is probably at 60 mg per mil, maybe even higher. Well, here it's still at 1 mg per mil. So you get local concentrations that in some cases will completely mess up your protein. And I've seen proteins indeed that once they've been through the concentrator and we went a bit too far, we could never monomerize them again. They were completely destroyed. On a gel, on an SDS gel, they look beautiful. But in real life, they're actually completely messed up. So although you think this is the best thing to do for my protein, maybe you don't have to run a gel filtration. You can run an analytical gel filtration to make sure you don't have lots of aggregates. But you don't have to run all your protein through a gel filtration. There's no need to if it's a well-behaved protein. And so when it comes off a Q column or any other column, just take it as it is and freeze it. You're done. You don't have to run it. Plus the time that you spend there. Obviously, in some cases, if you do have lots of aggregates, it's worthwhile doing it. But then you need to make sure that after you concentrated it, did you not introduce new aggregates? So you go through the job of getting rid of them, and then you make new ones. The other one, if you do have degradation products, in some cases, it's the only way to get rid of them, and not even completely. They might overlap, and then you may need to cut part of your peak. So that's another thing. I don't know if I'm coming to that. is that when you see a single peak, it doesn't mean that's a single protein. It could be a population of proteins that merge together into a single peak, but in reality they're not. So it's good to run a gel and really carefully examine what's in that peak. And you can do other techniques like multi-angle light scattering coupled to size exclusion to find out if you indeed have a monomeric species over the peak, or maybe you have dimers and monomers in that same thing. I'll show some examples later on. Just want to go through this very quickly. Buffer exchange, typically you dialyze it. It's a terrible practice. You spend this flexible tube and suddenly your protein falls over your bench. So you get the same results if you just simply dilute your protein 5 or 10-fold. If you come off a cube column and you want to bind to something, say a heparin column, and you need to go to low salt concentration, just dilute it and then bang it on the column, you're done. If that doesn't work, you can quickly run it over desalting columns. These are very rapid things that you can run at 20 mils a minute. You're done in no time. And if you need to change the pH, you don't need to go and dialyze it overnight. You might as well just directly add some hydrochloric acid or sodium hydroxide to change the pH directly onto your protein. Of course, you need to be careful that's within the buffering capacity of your buffer, because if you go, say, tris and you want to go to pH 5, by that time it's no longer good. And then storage. I think if you can freeze your protein, you should always do it, because you take a very important parameter out, or a variable out of your protein, is that's time. You stop time. So when you're able to freeze it, you can go away and pick it out of the freezer and it will be the same as it was a week ago, or maybe 2 months ago. If you store it in the fridge because you think your protein doesn't like to be frozen, then you have a time parameter, and maybe on the first day it's fine, and the third it's changing, and after a week it's all messed up, so you need to go and purify some more protein. Now, obviously, if you can't freeze your protein, that's the only way to do it. But if you can, then that would be the way to go for me. Now, freezing sounds easy. Just stick in liquid nitrogen and you're done. But there's several things that go into this. And one of them is that when you're freezing, it's a rather slow process. So if you look at your tube and you got, say, 100 microliter or even a milliliter, the outside will be frozen and the inside will slowly freeze as well. And during that time, you get all kinds of strange effects. One of them is that your buffer, some of them are very much affected by the temperature you're working at. So you're actually going through a pH gradient. And most proteins don't like to be at pH 5, so they will precipitate. And that's the reason why you then can't freeze your protein. So you can use PCR tubes, and there's people actually spent some time looking at this, and they found that if you use PCR tubes in small volume, you can get proteins to freeze that otherwise wouldn't freeze so well. The same as when you thaw it. Don't take it out of freezing and put it on ice, because you really prolong the period. Now your protein is going through a pH gradient, which it may not like. So it's much better to hold it in your hand or even on a warm water stream, so that you rapidly thaw it, and then you can put it on ice. But thawing it on ice is a very good way of precipitating your protein. And then, once you got your protein, it's worthwhile examining what the buffer actually is doing to your protein. And I will get back to that later, because you've chosen a buffer, probably tris because it's cheap and everybody's using it, but maybe tris is not good for your protein. You might want to use something else. You probably add some glycerol to it, but in most cases glycerol doesn't do a lot. And then finally, all right, let me move on. Yeah, so this is a protein solubility screen that we developed, and was based on an earlier publication by, I don't recall her name, but it comes back later. But basically, she found a way she designed a way of 24 different buffers, test them out, look for precipitation, and the ones that stay clear, you examine by dynamic light scattering, and that way you can find a buffer condition that is suitable. The way they set it up is rather large scale and manual, so I changed it a little bit to be robotic setup. And so they've got 24 different buffers, ranging from pH 5 to 10, and then it's without any additives, so you put your protein straight into that buffer, or there's a little bit of salt, or glycerol, or a combination of the two. And then you simply score your protein for precipitation or not, where you can clearly see when it's doing well or when it's doing bad. And this is very simple. It's already a pre-allocated crystallization plates, just like the other ones that we have in the basement. You use a robotic setup to put 100 nanoliter into 1 microliter lot drop, so you have a 10-fold dilution, and it's very rapid. And if you have high amounts of protein, 10 mg, you will get results within an hour. If you have lower concentration, or say 1 mg per mil, then you need to wait a little bit longer to see an effect. But this is a very good way. Why did it go wrong? Anyway, before the computer quest, I had a comparison of different results from proteins, and they are completely different. So some of them really don't like any low pHs, and they're all soluble here, while other ones are the complete opposite. So it's worthwhile testing this. It really doesn't take a lot of time. It doesn't like a lot of protein, and you may find buffers that it's very happy in. At the same time, you may find that your protein is actually stable over a wide range of pHs that you can use throughout your purification. If you want to bind it to an S column, in some cases you need to lower the pH to, say, 6. Wouldn't go much lower, but if you know that your protein can be happy at pH 6, then by all means use that. And if you can bang anything on an S column, it'll be very pure, because very few proteins do. Now, we also found that this is also useful if you want to do a particular assay. Say you want to do, in this case, we wanted to label our protein with a maleimide fluorophore, but every time we did it, it just completely crashed out of solution. So we did the reaction in the crystallization plate under these conditions. So rapidly, we found the conditions that would give us a fluorescently labeled protein that is still soluble. The same is for cross-linking. You can even take the whole plate with your protein and freeze it and thaw it. And yes, it's going to be different from how you do it if you do it in a vial, but it may give you an indication in which conditions your protein is happier than usual. So this is now coming back to what I alluded to earlier, John Carriage, who designed this 24 buffer system to first look at precipitation of your protein and then monitor it by dynamic light scattering. And this is an example of a bad protein, so you got a distribution of sizes that is more or less a single population, and then there's another group of higher molecular weight molecules that are clearly aggregated. And so by changing the conditions onto something that is a single distribution, you actually favor the protein for crystallizations, and that what they could show that indeed the ones that had a single dispersion would have a better chance of crystallizing. So it's again getting to know your protein, finding out what makes it happy, either by ligands or having the right buffers. And buffers, I don't mean just simply pH, because pH 7.5 in tris or HEPIS or phosphate might be having completely different effects on your protein. So it's not just about pH. Then protein characterization. Gel filtration is a really good way of looking at your protein. You don't need to run a 120 ml column to do it. We have 2.5 milliliter columns which run in just under 30 minutes, so you can quickly go through a whole bunch of conditions to find out how your protein is behaving. In this case, we're looking at a protein complex. These are 3 different concentrations of a protein complex, a dimeric complex. And although this middle one looks kind of single, and this is what I mentioned earlier, it looks like a single peak, so you'll be inclined to take everything because that's more protein. If you run it out on a gel, and in this case we quantify it, we find that much of the smaller protein is actually spread out over a large population, which means that this is the unbound protein and that's the bound protein, which in other words means that this is not a fully occupied complex. So we get some information out of this. You can do similar things with multi-angle light scattering. So as the protein comes off a gel filtration column, this is the UV trace. You also look at the total light scattered of a laser light, and that gives you information about the size of the molecule. Now, here it's an easy one. You really realize that that's not good, and you want to take this one. But you can also see that it's drifting slowly apart. So if you really want the cleanest protein for crystallization, don't take the whole peak, but maybe only limit yourself to this area. And recently we went to a synchrotron where they do the same, but now instead of using light, they use X-rays. And we used it for small angle X-ray scattering, and we get a similar profile. So now, rather than just looking at the intensity of the light, we look at the radius of duration that can be calculated. And you can see that this is a real nice mixture of different things, higher, middle, and then it falls apart even. So in this case, if we would have done the small angle X-ray scattering in the cuvette, we would have measured everything in one big mix, and the resulting data is a convolution of everything in there, which is not very useful. But by separating out on the gel filtration, we get much better data. And then the same holds for multi-angle light scattering. So getting to know your protein is extremely important to get to the crystals. Okay, so I briefly got this out of my notebook. This is an experiment I did a while ago. You can see about more than a year or so, a year and a half. And the reason is that I for some reason, I wanted to crystallize a protein that I already had the structure of. And when I solved that structure a while ago, I got crystals in many different conditions, and I was using a manual setup. So 1 microliter plus 1 microliter, you grease the cover slip, you bang it on. Well, very cumbersome, but I got many, many crystals. And when I set it up with the robotics tray, I didn't get any crystals in 400 conditions, and that puzzled me. So I went ahead and compared different conditions in which you crystallize stuff. So this is the volumes that you would use in the crystallization plate. I actually used to set the crystallization plate. And this is a bit larger volume in the larger plates, and that's the manual setup. And you can see that the different volumes, the way you set up, greatly affect how it crystallizes, and even reduces sometimes the chance of getting a hit. So these were actually multiples of the same condition. So here actually I did 4 times the experiment, actually 8 times, so one's at 10 and one at 20 mg per mil. So out of 8 drops, I only got crystals in 1. So that means that if you set up a crystallization series of 2,000 drops, you might well be missing many conditions because you're just not optimally screening. And my hunch is that by all means, go and do it. But if you don't get any crystals, then think about how you're doing it, and it might be worthwhile to do a few screens with larger volumes. And the reason for this is actually, there are multiple reasons. Is that, statistically speaking, if you have a drop size that's 10 times smaller than the one that you have, if you were to get 1 crystal in a 1 microliter drop, you get 0.1 crystal in a smaller drop, statistically speaking. So the chance of getting one are tenfold reduced. Then there's some other things that go into it. You have a smaller drop, so evaporation is going to go much faster. So that the time that you spend in the phase diagram, I'm not showing it, but the time that it spends in the area that's optimal for nucleation is much shorter. So again, your chances of getting that unique hit are reduced. And then there's another reason, is that typically in a hanging job where we use a larger volume, larger setup, the air in between your drop and your buffer is much bigger. And it takes a longer time to completely be saturated and therefore affect your drop. And again, you're slowing down the process of crystallization, which gives you more chances of actually getting a hit. Now, we all know that the crystallization facility works really well, but if you have a problem getting crystals, think about it and maybe set up a few trays by hand. And somebody, again, took the effort of looking at this, and they have a whole paper about how fast it is. And even the distance between, if you can change the distance, you can greatly reduce the rate at which crystals are growing. So it also helps if you have crystals all over the place and they go too fast and you want to slow them down. There's different ways of doing it. And one of them is simply making the distance between the surface of your reservoir and your protein drop, making it bigger, and that will slow it down as well. So now we get to freezing. I had a lot of cool things here, but they're all gone now. I lost it. But it's actually cryo-cooling, because we're not wanting to freeze it. If you freeze your crystal, what you get is ice crystals, and they'll diffract like crazy, and you won't see the protein diffraction at all. So what we're doing is cryo-cooling, which is trying to solidify the liquid around it in an amorphous state so that it won't diffract. Thomas Schneider has done a lot of research on this, but also Elspeth Garman and Jim Flugbraut. And I will update this presentation so you can look back and find some references that I had in it and I lost them. So that tells you something about the physics behind it. Why are we adding actually cryoprotectant to it? Why can't you just take your crystal and freeze it? Now, another good thing is, it's really a shame, we typically freeze them, and the reason is that the intensity of the X-rays that we shoot the crystals with at the synchrotron are so high that you would just destroy your crystal after 1 or 2 images. So we need to cryo-cool the crystals. But during the process, you might well mess up the quality of the protein, and there are different reasons for it. So it's worthwhile taking a native crystal and actually shooting it at room temperature. You can either do it in-house, you can also do it at synchrotron. It may last 1 or 2 images, but it will tell you how good your crystal was before you try to freeze it. And that means if you see a big difference between your native room temperature and your frozen crystal, you might want to spend some time optimizing it. And at the same time, freezing can also be a way to improve the crystal packing. So you can moderately dehydrate the crystal by adding more precipitant, and that will somehow rearrange the crystal packing a little bit, and that can improve the diffraction. Now, one note is that the improvement of diffraction you get is limited. It's maybe in order of half to 1 angstrom. So if your crystals are diffracting 30 angstroms, you're not going to get much improvement. But if they're 3 angstroms, half an angstrom resolution improvement is a big deal. Now, there's something called the free mounting systems, and there's other systems, and there are very nice ways. So you get your crystal not frozen, you catch it in a loop, and you put it in a humidified airstream. So it's set to a particular humidity. And then you put it in front of the X-ray, and you collect an image at that humidity. Then you change the humidity a bit lower, collect again, and you continue this until you see improvement of the resolution. And that's a more sophisticated way of trying to dehydrate your crystal. There's another benefit to it, is that for doing so, you need to wick away all the mother liquor that tends to be around your crystal. And by doing so, you do 2 things. One of them is that you get rid of the noise that the background, or that the drop gives, the amorphous drop, it contributes to noise, so you get rid of that. But at the same time, you can actually freeze crystals when they're completely on their own, when there's no mother liquor. Because all the trouble we go through in the cryo-protecting is about the mother liquor that your protein is in. If you can get rid of it, you can freeze any crystal without having to add any cryoprotectant. Well, maybe not any crystal. But if you were to do that by hand without a humidified stream, you will dry out your crystal, and that's not what you want. So you need to be in a special place where they have some way of having a humidified airstream. Or if you're lucky, I actually had, at some point, I had very long rods of crystals. They were too long for the loop. So you fish them out, and the majority of the crystals in the loop, surrounded by liquid that has been frozen in an amorphous state. And then a part of the crystal sticking out of the loop, nothing around it. And when I collect data on that bit, the data was at least half an angstrom better than the one that was in the loop. So you don't need the mother liquor, it's just a way for us to freeze them. But if you can get away with it, you get much better results. One more thing that I wanted to add is that when you freeze your crystals, you typically take a box of liquid nitrogen and you just stick it in there, and that's it. Now, when you do that, you have to be quite careful, because especially if you have a deep bucket, you might have an air, or a layer of cold air, before you hit the nitrogen. So as you plunge your crystal through it, it will actually cool down in a gradient, and you don't really do flash freezing. And between crystals, you get great variability. So it might be better if you fill up the liquid nitrogen all the way to the top. And the people actually have designed little motors that will blow warm air over that, so that what you get is actually real flash freezing. So it travels through room temperature air, and then it immediately hits the liquid nitrogen without having to travel through a buffer of cold air. Another way around it is that you freeze it in the gas stream. So if the cryo, what is it called? The cryo stream, which has a cold nitrogen gas at 100 Kelvin, is also a good way to freeze your crystal. And you can do that very simply. You mount your crystal in a loop, you put it on the goniometer, but you block the cryo stream, and then you remove it, and then your protein gets frozen in a cold air stream. So these are things that affect the way that you freeze your crystals, and ultimately will affect the resolution of your data. And then to end this all, so I'm not giving you a foolproof recipe of how to go from protein to crystal and good diffracting crystals. There are many, many, many different ways, and you need to find out for yourself what works for you. And kind of what I want to say is these results, the first structure of DNA and the first protein structure, were not solved because people did the same thing over and over again. But they were solved because they did something that nobody else has done. And the same is probably for your case. If you have a difficult project, doing the same as everybody else is doing is not going to make you be the winner. You're only going to be the winner if you do something that's different from anybody else. And with that, if there's any questions.
Thank you. In short, be creative. Yes?
Exactly.
Okay, any questions?
Okay.
It was silent. At least one question. Yes, that's good. Yes. You are the winner.
In comparison to dynamic light scattering, how would you compare using thermal shift as a way of screening buffer conditions for solubility?
Thermal shift?
Yeah, so using a thermophore to see how stable your protein is in the buffer?
Oh, and then you heat it up, right? Yeah, I know about it. Well, it is somewhat nice, but you need a good machine to do it, and not everybody has it. And when you do have it, it's being used by other people, so it's not something that can easily accessible. The other difficulty is that as you're changing temperature, you're also changing pH. So what are you really looking at? Are you looking at the buffering capacity? So phosphate is extremely temperature sensitive, when HEPAs might be a little bit better. So at the same temperature in phosphate, you're already at pH 9 or whatever, well, and that will precipitate protein, while the one in HEPAs is not yet. So it's not a real fair comparison, I think. The way that we've set it up is a bit nicer because you can do it actually under the condition that you work with your protein. You can take the plates at room temperature or at 4 degrees, whichever you like.
Any more questions? Yes.
Yeah.
Can you do dynamic light scattering on membrane proteins at all?
There should be no real reason why you couldn't. If they are in different states, then you will get a messy result. But that's also for normal proteins that are in a solution and not membrane bound. So if you have a detergent that keeps your protein soluble and it gives a nice cover around it, that's fine. If you need to have a detergent concentration that will also cross micelles to exist, then of course the result is going to be a mess because the micelles will scatter enormously as well. So you need to find that concentration where it's just protecting your protein but not causing any micelles. Yeah.
Just a question about this, the freezing of the crystals. Freezing crystal sounds to me like a total oxymoron, but
It sounds like what?
It's an oxymoron. It's like military intelligence or something.
Mm.
Like the crystal's already frozen. You can't freeze a crystal. So it's all about freezing the liquid around it. So
Yes.
how much does it matter whether or not that's crystalline or not in this case? Because if you really wanted to make it amorphous, then there are ways of doing that very carefully that we use for cryo-EM. But I guess you don't do that in crystallography. Is that right?
Well, we try to make it amorphous by adding cryoprotectants like glycerol or MPD or PEG 400, which, if you do it fast enough, they prevent cubic ice of forming in your drop. So yeah, it's a brute force approach. But as I said, if you can get away with just removing the complete mother liquor and you freeze your crystal that way, that's fine.
So does anybody ever just sublime it off? So after it's already frozen, just sublime away all the liquid phase, and you're left behind with the crystal.
Well, you can do that if you have a humidified chamber, because if you sublime it off, then you also take all the water out of your crystal, which would then mess it up. So when you use these systems where you can have a humidified airstream and you can go at different humidities, what you find is that there may be a sweet spot, but if you go over it, you completely ruin your crystal. And in some cases you can go back, in other cases you can't. So it is a very careful process, and there's a lot of improvement to be made, that's obvious. But for now we can just get away with it. In most cases it works. If you just add 15% glycerol, it will work. So why change it? But there are cases that you need to improve it, because 3 angstroms might just not be enough. But if you can get to 2.5, that's a big deal.
Water's 3.7 or something, right? So that's where you get in trouble if you go below.
Yeah. Plus, the cubic ice might also destroy the crystal lattice because it's not the same, which then again deteriorates the quality of the crystals. So when you take a native crystal, or a room temperature crystal, and shoot it, and you compare it to one that's frozen, you'll find that they're different. So the freezing method is not optimal by far, but we can get away with it in most cases. Yeah?
To follow on from Chris' question, and has anybody tried without any cryoprotection, just freezing in a vitrable like we do with our cryo grids? What happens?
Well, that's what I was saying. If you can get rid of the mother liquor, because it's the mother liquor that's the real problem. So the maybe I'm not making it clear, but oh,
A little bit translation, yes.
Okay.
That lecture about translation rotation was last time.
So if you zoom into a loop, and you're tiny, you can hardly see it by eye. There's a nylon loop that affects very poorly. And then there's a loop, and it can be of different sizes, and so you catch your crystal in there. And then around it is the liquid that your crystal is in as you pull it out. And that's the problem. Nowadays you can get really cool loops that are made like a tennis racket. of a material that diffracts X-ray very poorly, so they don't leave any background. And so now you can put your crystal on there, and then take a small paper tip and try to remove all the area around it. And then you can freeze it, like your VitroBot. But if you do this just in any room, then you have a very short period where you have the sweet spot, and then when you move into the liquid nitrogen, you dehydrate your crystal even further, and that makes it possibly even worse. So you would like to do this under a controlled environment in whatever box that has a particular humidity. Like the VitroBot, yes.
But one important difference is the size of the object. Magnetic crystals are much bigger. So it actually turns out that small crystals freeze much better than large crystals.
Yeah.
Another thing also that I think cryo don't really worry too much about is the tiny change in volume upon freezing. So when you do this in the loop, actually the biggest problem is not to stop it, the ice formation, which is trivial. The biggest problem is that you need to have the change in volume upon freezing needs to between the liquid around it and the crystal needs to be similar. Because if it isn't, you will destroy the lattice of the crystal, which you detect very easily.
That's like a 1% change, right?
Yeah, but that's enough. So in cryo we don't worry about this because you don't know that this happens. But in crystals you can see it happen, and you get very often distortion of the crystal lattice.
Another thing maybe.
So actually the difficulty in finding a cryo buffer is that you need to find a condition that both the crystal and the liquid surrounding it have as little differential in volume change.
So on that note, we have X-ray equipment or X-ray machines downstairs, and they're being used less and less frequently because everybody goes to the synchrotron. But they're actually great for testing cryo conditions. It's a bit of work, but it's definitely worthwhile. So try and freeze your crystals in many different cryo conditions. Try also increasing the amount of your precipitant, because that may dehydrate your crystals a bit. And then you might find a condition that you get slightly better results before you even go to the synchrotron.
Any more questions?
Have you heard of proteins purified when freezing it to actually drop the actual purified protein directly into liquid nitrogen as opposed to putting it into a tube?
Yeah, that's certainly a possibility.
Wouldn't affect the protein too much?
No, not at all. We actually done it a little bit. We were shipping samples to Berkeley, and for some reason, and that was the way that we froze our protein drops in liquid nitrogen.
Small spherical balls.
You know, everything that makes it more cumbersome to do anything, I try to avoid. So if that's the only way to get your protein frozen well, and it will be faster because you don't have a plastic wall surrounding it, that if you need to, absolutely do it. But if you can get away with it, and therefore you can do something easier, I would choose the easier method. But the PCR tubes, they are thin-walled for PCR, but you can also use them for the same reason, for protein freezing. Yeah.
Any more? I have one question or two comments or questions. What do you think about slow cooling? People are using and needing. I've heard that some people have used that. What's your opinion on that? I have no opinion.
Is that where, do you refer to when you freeze your crystal and then thaw it and then freeze it again?
Slowly just decrease the temperature or something like that. They call it annealing.
That sounds to me like another thing that you could try, but it's certainly not a guaranteed method to work.
Yeah, it doesn't guarantee.
So one thing that you can try the synchrotron is that if you got crystals and it don't very well, you can briefly block the cryo stream so that your crystals will thaw, and then you immediately freeze it again. And if you do that a few times, there has been some anecdotal evidence that may work. Yeah, but
yes.
When it's in the beamline, it's there anyway. You might try it, you don't lose anything.
Yeah, sometimes dehydration increases resolution.
Yeah.
Not always.
Yeah, not always. No.
It's usually worse.
Well, I must say, I had a case indeed that.
You want one crystal. Maybe for your case.
Yeah. So this crystal, by the way, I could never. These are so big that I couldn't cryo cool them, because when you put them in the cryo buffer, they would just crack. So big crystals are not always delusion, and Jan already mentioned it earlier. They're actually very tricky to handle because you get such temperature differences from one end to the other and osmotic differences. So they look very pretty, but they were not very practical, and I had to go to smaller ones to get good data.
Okay, thank you. Thank you.